I'm going to write them up and we'll go through each one individually. All right, here they are. So cytosol, that's the first thing we'll talk about. What is the cytosol? It's that fluid part. So we say cytoplasm is this gel-like fluid, so the cytosol is the fluid itself. Next we can go to the cytoskeleton. So the cytoskeleton is made up of three different things. We've got microtubules, also called filament tubule. We have microfilaments. And we have intermediate filaments. And let's put these in order from biggest to smallest. So the smallest is going to be a microfilament. Then we'll have intermediate filament. And then we have a microtubule or filament tubule. What is a microtubule? So the cytoskeleton, it's a bunch of proteins, tubules and filaments. They're going to add integrity and structure to the cell. Okay. So a filament tubule, it's hollow. It's a hollow protein. Gives the cell structural integrity. And it also helps distribute organelles around the cell. So we did microtubules, or these filament tubules. They're the biggest. They're hollow tubes made of proteins, deal with structure and integrity, and they distribute organelles around the cell. In the middle, we had those intermediate filaments. And all we're going to say, tough, insoluble fibers that add strength. Okay. Microfilaments, super important. So if you remember with endo and exocytosis, here's my cell, and I want to bring in compound A, so I make this little divot like this of my plasma membrane, and then I surround it. Sorry, going a little too fast. So I want to bring in compound A, so I bring it in through a vesicle, and I attach these parts of the plasma membrane, and then I get my vesicle inside. So let's draw that. Now I have my vesicle inside. So this is what we saw before, compound A. But the real question is, it's not like this plasma membrane is just magically, this folding in thing, it's called invagination. Okay, so this plasma membrane isn't just invaginating normally. It has to be acted upon to do that. So let's put compound A back outside the cell. And now let's add, right underneath the plasma membrane, let's add some microfilaments. Okay, now let's contract those microfilaments this way. Well, if I contract these microfilaments this way, it's going to pull the plasma membrane in, and that's going to start that invagination. And then the plasma membrane will eventually fuse. So you're going to keep pulling, these microfilaments are going to keep pulling, and eventually it's going to pull down so much that these sides are going to get closer and closer until they touch. So this side was here, then it was here, then it was here, until eventually this side here, here, and here, fusion of plasma membrane. So this is how microfilaments are working for contraction to pull in things for exo and endocytosis. We'll say add structural integrity. And remember, they're located just beneath the plasma membrane. Alright, so in quick recap, we did the microtubules first, then we did these intermediate filaments, and then these microfilaments. So those are all components of the cytoskeleton. They're these proteins that help add structure and integrity to the cell, and they also have to do with that, like the microfilaments pulling stuff in and whatnot. So this is the cytoskeleton. The cytosol was that fluid. So if we go back to the cytoplasm, that was what we were originally starting with. We've done the cytosol, we've done the cytoskeleton. Now we need to look at cellular inclusions. So let's put a topic, cellular inclusion. So cellular inclusions are things stored in the cytoplasm. And he gave us a few different examples. So glycogen is a cellular inclusion in muscle cells. Triglycerides are cellular inclusion in adipose tissue. Melanin is a cellular inclusion in melanocytes or skin cells. All right, those are all good. So all three of these, which is glycogen, triglycerides, and melanin, these are the cellular inclusions. All right, talking about a cellular extension. Cellular extensions. So this is different than those finger-like projections of the plasma membrane, which were microvilli. This is something other than the plasma membrane coming off. So one form are cilia. So we have two types. We'll do cilia. My favorite example of this is PCCE, so pseudostratified columnar ciliated epithelium. And what we're doing with that is we're going to draw a few cilia coming off the sides of it. Boom, 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 boom. So these are found in the throat a lot and the respiratory tract and whatnot. So that chemical gets down into your lungs where you have this pseudostratified columnar epithelium. And what it can do is it can trap this toxin, and these things can move. So cilia they're motile. They can move in this direction or they can move in this direction. And they're not moving, they're waving back and forth, right? They're making these, just flap your hand back and forth. I can't show you, so just move your hand back and forth, and they're able to move things in certain directions. So cilia, you'll find these all over the place, but again, trapping dust or pollen in our respiratory tract, sweeping that towards the throat to be coughed out. We'll also find cilia in the female reproductive tract, because after you deposit the sperm and it starts traveling up towards the fallopian tube to become a zygote, to make your baby. Well, as that sperm is coming up, we have these cilia that can kind of wisp the sperm and egg together. We'll get to that next semester. So that is one extension, the cilia. The other extension we want to look at is a flagella. The only cell that has a flagella in the human body is a sperm cell. So here's the sperm cell, and here's its flagella. So this flagella is used as a propulsion mechanism. Boom, boom, boom. Propulsion, which is movement. So this thing whips back and forth like this, and it sends it in that direction. That is a flagella. Remember, only one in the human body with a flagella is a sperm cell.